Okay, next we have Dr. Lisa Trevino. She serves as the Vice President at the DHR Health Institute for Research and Development. Dr. Trevino earned her Master of Science in Molecular Biotechnology from the University of Texas San Antonio and a PhD in Molecular, Cellular, and Developmental Biology from the University of California, Santa Cruz. Before joining our team, Dr. Trevino gained research experience at St. Jude Children's Research Hospital in Memphis and at the Human Genome Sequencing Center at Baylor College of Medicine. Dr. Trevino's areas of expertise are in cancer genetics, genomics, and pharmacogenomics. Welcome, Dr. Trevino.
Okay. Thank you, everybody, and welcome. It's a pleasure to see so many of you. Thank you to the teachers and to the sponsors that brought the students out here to listen to some tremendous speakers. There's a wealth of knowledge that you're going to gain today, and Dr. Rao has pressed upon the importance of biomedical research, clinical research, how that affects our community, and you can take that and internalize it and see how perhaps it may affect you and perhaps your own family members, whether it be if they have diabetes or cancer or liver disease, obesity, or even if they may have been affected with COVID. Always know that you've got physicians and scientists, not only here locally at DHR Health, that are working to treat your family members, but the scientific and medical community at large, all coming together to help us get these cures, these vaccines, in order to prevent these diseases. So my talk today is going to stress upon what is referred to as personalized medicine. And you'll hear for the remainder of the talk how I am transitioning from personalized medicine to more what is called precision medicine, okay? And how we are using precision medicine to better treat you specifically for the disease that you have, to help you get a better outcome and maybe not suffer many of the side effects or consequences of that disease. So throughout the talk, I'll talk to you about what precision medicine is, what are some current applications of precision medicine, the history and examples of precision medicine, where we started almost 100 years ago to where we are now, with guided therapy for diseases such as cancer, and in particular cancer that has made a most lasting impression in the field of oncology. And at the end, how does precision medicine affect you and the future of your healthcare? So precision medicine, it's really a broad term, but it's healthcare that's tailored to you. So I'm assuming a lot of you have been in quinceañeras or weddings, right? And so when you're in those types of events, what do? Girls, you go and get fitted, right? You go get fitted for a dress. Guys, you go and get fitted for your tux. And what are they doing? They're making sure that it fits you, that it's not too long, that it's not too short, that it's not too tight, too big, etc., right? So really this is what the term precision medicine does. It's healthcare that's going to fit you, and it's going to be tailored to you. It's a form of innovative medicine that uses information about a person's own genetics to prevent, diagnose, and treat. And I want you to pay particular attention to the order in which I put those words, right? Right now, and maybe within the last 10, 15 years, or maybe even before that, medicine has been all about treating the disease and treating the disease and treating it. Well, why don't we step back a little bit and learn how to better prevent it, diagnose it, and then treat it, right? So the idea now is to move and transition away from the arena of treatment to, you know what, let's see how we can earlier detect the disease, better diagnose it, and keep us from getting those diseases. Not only does precision medicine involve your genetics, but it also has to take into account the environment that you were raised in, that you live in, the environment that your parents were raised in and lived in, but also, like Dr. Cordova-Kissy said, the lifestyle that we live in. And again, using all these factors to better and more accurately and effectively treat the disease that you may have. So not only is precision medicine aiming at better finding a treatment for you that's going to help you better, but it's also going to help us avoid you having secondary effects, help you avoid having to take those extra tests that you don't need, and something that may even be harmful to you, right? So it's a two-pronged approach with precision medicine, identifying the best treatment to help you, but also to protect you from hurting you. So everyone here, you're what, sophomores, right? Everybody here is a sophomore, you've taken biology, so I'm sure at some point throughout your courses and classes, you've learned about DNA, right? And the importance of DNA. Well, today I'm going to dig in a little bit deeper about how important that DNA is. So in precision medicine, DNA really is the main player. You may have also become familiar with RNA, right? And protein, and a lot of that and how it goes from DNA to protein in your classes. So DNA really is the material that every single living organism has, and it's information about how you will look and how you will function. So essentially, it's all our genetic blueprint, right? So you've all seen maybe blueprints for homes, blueprints for new buildings, and they've laid it out and said, okay, this wall is this tall, this wall is going to be this wide. So our DNA is our genetic blueprint. The DNA in humans will determine something as simple as the color of your eye, maybe the color of your hair. But the DNA that we have in us is also going to determine how we process medications. We've talked to you a little bit in brief about some of the studies that we have that tell us what patients have certain areas in their DNA that's going to allow them or not to process medication. And also, in our DNA, in our genetic blueprint, lies whether or not we are susceptible for certain diseases, cancer, Alzheimer's disease, Parkinson's disease. In your classes, you must have seen that there's 4 bases that DNA is made up of. It's A, C, T, and G. All of those will combine in over 3 billion bases throughout the human body. Our human body has trillions of cells, and in every cell, you have a combination of those 4 letters. So just imagine, right, how that combination is in our body that's circulating through our heart cells, our liver cells, our brain cells, our hair cells, all has DNA, 3 billion bases of DNA floating around in us. In that DNA, in that long stretch of letters of A, C, T, and G, it's formed into specific words, like I like to call it, or sections or segments in the DNA that tell you, okay, this particular segment is going to detect eye color. Well, in that segment, there's a gene, right? So you're all familiar with genes. And in the human body, or in the human, Homo sapiens have between 20 and 25 genes, 25,000 genes. So from all those trillions of cells and all those billions of base pairs, we've narrowed it down to about 20 to 25,000 genes. Now here's something to think about. The fruit fly, Drosophila melanogaster, maybe you've heard it in your classes as well. That little itty bitty fruit fly has 14,000 genes. So how is it that we just have relatively few more genes than the fruit fly and look at the complex and beautiful model organisms that we are compared to the fruit fly? So that's just for consideration and to see the complexity of the human body, the complexity of what we are made of, and how it gives rise to us functioning as healthy as we can be, or unfortunately, how sick we may be. So, like I just mentioned, if we were just to kind of take stick figures of all of us and lay it all out, this is what we would look at, right? Technology now today has allowed us to be able to read out every single base that we have in our human genome. So we're walking around with all these different combinations of letters all across, each and every one of us. Now there's areas, and for the most part, we are all very similar in our DNA, but then there is uniqueness to each and every single one of you. Dr. Rao mentioned earlier today how there's differences in our genetic makeup between Hispanics and Latinos, African Americans, Asian Americans, and even those Latinos that have a Native American ancestry. There's differences in different regions of the gene that make them more susceptible to a disease or more likely to be responsive to a certain medication. So keep this in mind, right? We're all really just walking around with really functional information in our DNA that's going to really lead and guide how we're going to live long, healthy lives. So a quick fun fact, like I mentioned, every single cell has about 3 billion, our human body has about 3 billion bases. So if we were to take the DNA in each and every one of our cells and stretch it out, completely stretch it out, it would go to the sun and back 300 times. So that's a real long way, right? So imagine that the DNA filled every single row, every single aisle here, every single seat that you're in right now is a species of DNA, right? And in that, you've got specific genes that I was just talking to you about. Research and science and medicine today has come about that we have now been able to really read out the entire length of the human genome and identify these key genes that may be affected or associated with specific diseases. So think of that, super, super, super long. However, we have it in the palm of our hands, the identification of genes and areas in our DNA that's going to lead to precise and precision medicine, like I was talking to you about, to help better treat you for the disease that you may have. So here is kind of some history. There's a lot of detail here, but let me just tell you that DNA was first discovered in the 1800s, and it was discovered in some pus-filled swabs by a scientist by the name of Friedrich Miescher. So back in 1869 to 1953, that's about 90 years, close to 100 years in between, in which we were able to identify that there was something in that pus, to actually looking at what DNA and how it is structured. So Rosalind Franklin, together with Watson and Crick in 1953, determined, wow, it's shaped in this double helix that you've all seen in your textbooks. And in that double helix, you've got the pairs of the A, C, T, and G. So all that research that transpired between the 1800s and 1950s then took us over for another 50 years to when the completion of the entire readout of the human genome was completed. So again, what I really want to depict in this time frame that you see here is that much research has been done, much investigation has been done, clinical trials have been done to determine really exactly how our DNA is laid out, how is it different from me to you, to your cousins, to your siblings, unless you're identical twins, and even identical twins don't necessarily have the exact same, they've got about 99.99% of the same identical DNA. But aside from that, no DNA is alike. There's similarities, and then there's very big differences. So much research has been done to date to be able to allow physicians, scientists, whomever it is that's helping better treat you, determine this is how we need to treat you. This is how much medication we have to give you. These are some key milestones in precision medicine. Dr. Rao, Dr. Kissy mentioned how these new medications are targeting specific genes. So in the area of breast cancer, we've identified over 100 genes that are either associated with what type of breast cancer a woman or a man may have. We've been able to pinpoint exactly that particular gene in the genome, working together with companies that make drugs and compounds to be able to target that. And so we've come a really long way from treating patients, like they say, with a one-size-fits-all type of approach. So precision medicine has taken us from really casting a really large net to treatment to being very specific. The same thing for lung cancer. Hundreds of genes have been identified that if a patient comes in and you're able to identify that they have mutations or errors in these particular genes in their DNA, we have a quicker and more effective and cleaner way of treating them. Hyperlipidemia, which is really elevated cholesterol. I'm sure a lot of you know someone that may have any one of these diseases, cancer, breast cancer, lung cancer, or someone that's taking medication because their cholesterol is too high. Again, precision medicine has allowed us to target specific genes, treat you better and cleaner so that you have a better outcome, so that you're able to better respond to the medication. Alzheimer's disease, I'm sure we know some of our family members, grandparents, aunts or uncles that may have dementia or Alzheimer's. There is a lot of advancement also in us identifying regions in our DNA and in our genome that is specific and associated with Alzheimer's. And what does that mean? Well, we have an area to focus on, right? That we're no longer really searching and really fishing in a deep, wide ocean when we have genes, and precision medicine has guided us to better treat and locally treat and help maybe cure these diseases. Dr. Rao has made really good points throughout the duration of our talks today that not one particular disease has a definite cure. Research is really contributing to us to identify more, direction, better direction for a cleaner, less kind of effect or side effective type of approach to treating these diseases. So again, precision medicine is something that we've come a long way, and it's really through the identification and sequencing of a patient's DNA, our DNA, to identify areas in the genome that are associated with diseases. It takes into consideration your genetics, your environment, your lifestyle, and again, tailoring it towards you and specific to you. We can no longer treat every single disease as a one-size-fits-all. It's just not going to work. We've seen that. We've seen that, and I'm sure you've known a patient that has cancer, and you see that they're very sick, they lose their hair. A lot of the side effects of chemo are being mitigated now by precision medicine because we have cleaner, more direct types of treatments, and all through a result of precision medicine. So what are the tools? How are we actually identifying these genes and the patients that come to our clinics? Well, it's gotten to a point where it's as simple as a blood stick on your finger. You can take a blood from just a pinprick, you can extract DNA from that blood, and then you can read out the genome, or you can ask specifically, let me see the sequence of the genes that we know that are associated with cancer, or lung cancer, or Alzheimer's, or obesity. So it's as simple as that, right? We're trying to move away from biopsies. So biopsies is actually an invasive procedure in which a physician will go in, you'll have to be sedated most of the time, and they'll take a piece of the tissue that's affected, whether it be the liver, or your breast, or the prostate, or the pancreas, and then they too there will look at these biomarkers in that piece of tissue and determine whether or not you've got the cancer, whether or not the cells appear different, whether or not you have the genes that are mutated in that biopsy. But that's quite invasive, and a lot of the work that we're doing to date, and even the work that we're doing at the Research Institute currently, that Dr. Betancourt is leading, myself, Dr. Rao, and others, is we're trying to move away from this invasive biopsy approach to what's called liquid biopsy. In other words, we can now move from doing that invasive procedure of a biopsy to taking a blood draw, or that pinprick that I mentioned, and looking to see whether we can match what we would have found in the biopsy to what's in that blood draw. So what would you rather have, the biopsy or the blood draw? What was it? Exactly, right? You go in, it's quick, it's fast, it's relatively harmless, the risk is a lot lower, and we can tell you the same thing as if we would have taken the biopsy. So a lot of research is going into validating these liquid biopsies, and now what they're calling it is liquid gold. And you can find the cells that are circulating in the blood early on, right, to see whether you're going to have a predisposition to any one of the diseases that I mentioned. So precision medicine has taken us from sequencing out the entire genome to identifying a handful, maybe now with hundreds of genes, but now we're trying to work to identify a handful, maybe 2 handfuls of genes that we know that are associated with specific genes so that we can better treat you, prevent less of the effects that we know that's associated with several medication. So what does this mean for you and your healthcare, for you and your family? Well, faster and earlier detection of certain markers. Sometimes patients, especially those patients that are unfortunately diagnosed with pancreatic cancer, that one is asymptomatic. You come in at the very end and you determine, oh wow, I have stage 4 pancreatic cancer. Well, what if we were able to detect that very early on by a simple blood draw? That's where we're moving, so that we don't have to wait to the very last minute to tell a patient, you're at really high risk of getting this type of cancer. It's cleaner. It's safe for treatment. Like I mentioned, chemotherapy, trust me, I know. I had a niece that had leukemia. And I'm telling you, it's true when they say, and I don't mean to be as harsh, but the chemotherapy will kill you before it cures you. Because it really is something that's taking everything all at once. It takes the good cells and the bad cells. And what precision medicine is doing is really just helping us target those cells that have the cancer. You don't have to take the neighboring cell that doesn't have cancer. So if we can get to that, then I think we're going to be a lot better as a healthy and healthier society. Potentially less side effects, which is what we all want, less time in the hospital, and really, really important, it's going to be less expensive, because now we're not just completely throwing, like they say, the kitchen sink at trying to treat you, because we know exactly where to go, where to treat, how much to treat. So precision medicine has brought us a long way. I think it's going to be even way more advanced in the next couple of years. And I think even in the arena of COVID, we're going to be able to identify those patients that are going to respond to medication, to treatment a lot quicker. Cancer, we know for sure, is going to be all about precision medicine. This is just a quote that was stated by someone very important back in 2015 when the Precision Medicine Initiative was kicked off. So really, we've always known that you're unique, that your family is unique. Now we know even better how unique you are, how unique your DNA is. And the fact that we're able to deliver the right treatment at the right time to every patient is all we all strive for in the medical community. So thank you for your time. It's now lunchtime. I'll take any questions if you have any. So thank you, everybody. Enjoy the rest of the day. If you have any questions, Dr. Betancourt's always around, Dr. Rao. All of us are here to help you. Thank you very much.